Hello, my name is Jonah Cullen, and today I'm going to talk about my project from the genetic code to the horse, how translation of DNA to RNA differs between tissues and why it matters. A fundamental endeavor in genetics is associating a phenotype or observable characteristic to genotype, and this usually occurs in one of two ways. With forward genetics, we start with a phenotype of interest and try to uncover a mutation associated with it, or reverse genetics, where we start with a mutation and try to work to figure out what the ultimate phenotype will be. As you might imagine, in animal medicine and genetics, we're talking about forward genetics, where we again identify a phenotype or disease of interest and try to find a mutation that is associated with that. Normally, DNA is transcribed into RNA and ultimately translated into protein with a normal function. A mutation may disrupt this process through multiple ways. One being it could just alter the function of the final protein product, if a protein product exists at all, or it may alter the expression of the RNA and ultimately the protein. And this can occur in a tissue-specific manner. So it might be one level in the liver, different in adipose or fat tissue, and then yet different in heart as well. So the question is, how do alterations to normal expression cause this disruption reflected in the phenotype? And as all the cells in the body contain the same DNA, you might want to first ask, how does one cell become a liver cell and another a heart cell? And this is through gene expression, where certain genes are turned on or off to become a, for example, hepatocyte and ultimately form the liver. Or similarly, a cell destined to be part of the heart will have a different set of genes turned on and forming a cardiomyocyte and ultimately form the heart. Thus, capturing and characterizing normal gene expression in a tissue-specific manner is a key resource in linking genotype to phenotype. And that is exactly the objective of my project, where we wish to catalog the tissue-specific expression in horses. And we'll ultimately improve two things here. One is the physical annotation, so that is identifying where genes are located in the genome. And then two, the functional annotation. And this is the concept of assigning or predicting the function of a given gene and what it may ultimately do within the tissue. Towards that end, we use RNA-seq, where we extract RNA from all of our tissues of interest. We run them through the analysis pipeline that we built, and we ultimately end up with this sort of table describing the expression of each gene across all of those tissues. So this is the quantification aspect of this. And what we can do with that then is start asking some questions. And one of the first things we can see is that tissue type is the driver here. We can see here the samples are grouped by the tissue type, meaning that the genes expressed are more similar within tissue than between. And this makes sense based on the knowledge that it is gene expression that is driving the differentiations of cells to these specific shapes, functions, and ultimately tissues. Additionally, we can leverage what are known as co-expression networks to predict the functions of genes with unknown function, for example, expressed in adipose tissue here. Or we can identify genes specifically expressed in the liver. And this is important because if we are looking for a mutation that causes a disease of the liver, we might start with this list of liver-specific genes first, or rule out other genes that are not expressed in the liver. The major impacts of this project are, one, this will serve as a catalog of the normal tissue expression in the healthy horse. Two, as I mentioned, we improve both the physical and functional annotation. And then three, this will enable researchers to more accurately prioritize their mutations associated with a phenotype or disease of interest. And I want to thank you for listening, and also want to thank some of the people highlighted here.